Hello, and welcome to the CAP Today webinar for Wednesday, July 20th. I'm Bob McGonnagle, the publisher of CAP Today, and I'll be your host and moderate a question and answer session after our formal presentations. Today's webinar is entitled Methylation Analysis, a new approach to sarcoma classification. Today's webinar is made possible by a special educational grant from Illumina. We want to acknowledge that important support and thank Illumina for their enormous help in allowing us to bring you this very important program. Our speakers today, and I'll say more about them in due course, are Dr. Andreas von Deimling and Dr. Christian Kölsche, both from Heidelberg University in Germany. Each of these gentlemen is in Germany now in the early evening and joining us. And we want to welcome them. As I said, we'll say more in a moment. But before we begin, I just want to go through a few housekeeping items that I think can enhance your enjoyment and appreciation for today's program. We suggest that you may wish to refresh your browser. Also, please notice in the lower right-hand corner of your computer screen, you'll see a number, and we have live help available for your call. You can call 858-345-5916, and you'll get live help for any kind of technical issue. Also, below the slide box, as you see there, we have a section labeled Questions and Answers, Q&A. If you have any technical help with audio or the website, you can also type in that information, and we'll have helpers live all throughout today to help you with that. Of course, the question and answer box is primarily intended for you to type in your questions or type in your comments. You can do that at any time during today's webinar. As I said, we'll follow up with those questions and comments at the end. We hope to provide you with the full slides and audio of today's webinar in about a week, and you can see that and read that at captodayonline.com. You're likely to see some follow-up email from us and perhaps from our sponsors, so I wanted to let you know that. It's important, as always, that you know that CAP Today does not endorse any product or service that may be mentioned today in our webinar. And, of course, any comments that I make are purely personal and not to be taken as policy of CAP Today or the College of American Pathologists. Finally, I've been asked to let you know, and I'm very, very glad to call this out. In CAP Today in November of 2021, we had a very important cover story. Dr. Dan Bratt, a distinguished neuropathologist actually at Northwestern, where he's the chair here in Chicago, was on the cover, and the feature story was the sizable shift in CNS tumor classification with the new WHO classification guidelines for central nervous system tumors. And of course, the molecular importance of that is worthy of calling to your attention as you begin today's webinar. Let me introduce the two speakers, and then I'll ask them to begin. Dr. Andreas von Deimling is in the Department of Neuropathology, the Institute of Pathology, and at the University of Heidelberg. He's also affiliated with the Clinical Cooperation Unit in Neuropathology of the German Cancer Research Center. Very important. Dr. Christian Kölsche is within the Department of General Pathology at the Institute of Pathology at University Hospital Heidelberg. And with that, I will invite them to begin.
Hello, this is Christian.
And Andreas.
And we both have decided to split up our presentation in two parts. So Professor Andreas von Deimling will begin with the introduction, and I will cover the middle part of our presentation, which primarily deals with sarcoma classification. And with this, I guess we can start.
So hi, Andreas von Deimling speaking. I will do the basics. Basics includes this slide, disclaimers. I'll give you 20 seconds to read this properly. I think 10 seconds should be enough. And then I'm starting to introduce you to the principle of methylation-based classification. When I said basics, you see basics is simple. Look at this graph here. We have two axes. One is proliferation, one is differentiation. Every cell has, at the beginning, let's start with a fertilized egg cell, a zygote, has a high proliferation but a low differentiation status. And over time, we have very different cell populations, organ-specific cell populations, which are highly differentiated, but which are decisively lower in proliferation. So we move somehow up here. In order to do so, the cell has to employ different genes. As we know, we have 20,000 genes roughly, but they are not all always operational. And on the path to differentiation, of course, we have to shift through a large array of different genes. And the genes active here at the beginning of the single cell status, genes active here, is a very different set of genes active up here when we have a terminal differentiated cell. So what happens on this path? Well, genes have to be turned on and off, and this is done partly by regulating gene promoters. Gene promoters are in front of genes, and they have so-called CpG sites, which play a major role in activity of promoters. If CpG sites are methylated, the gene is silenced. If they are unmethylated, the gene is active. Very simplified way. So we have here a constant demethylation and methylation of promoters during this path of differentiation. Meaning here this cell has a completely different pattern than the cell being positioned here. And this is very important because this pattern of methylation relates to a highly specific differentiation stage in a distinct differentiation pathway. During this path, the cells go through, something can happen. Let's say here, EVSR1, FLY1 translocation. This is a tumor-initiating event, and then, if we are unlucky, we develop a tumor, in this case in Ewing sarcoma. One thing is very crucial. At this very point, the cells do not further differentiate, because if they further differentiate, it would not become a sarcoma, but a terminally differentiated cell. So the methylation profile, roughly speaking, is frozen at this point. And then it stays frozen throughout tumor progression. Lower grade, higher grade, very bad, very high grade. But the methylation profile does not change significantly, which means that if we look at the methylation profile of a tumor, what we really do is we look back into the cradle of the tumor. We are looking back to the latest normal cell population and the methylation pattern which was present at this time. And this is the reason why methylation-based classification is very well suited for classification, but it usually, and in most instances, fails at grading. Grading is something where progression plays a role, the additional changes which occur during progression, these are not reflected in the methylome of the tumor cell. So, essentially, we are looking at one axis, which is for classification. The other axis for grading would probably be better addressed by by next-generation sequencing and looking for fusions or tumor mutations. I have made a pretty bold statement here, saying that the differentiation, the methylation pattern is quite stable throughout tumor progression. This is very hard to prove, but I can give you a practical example. And as this technology was developed for brain tumors, this example also comes from brain tumors. What you're seeing here is an unsupervised clustering of tumors, so-called ependymomas. There are different kinds of ependymomas, the posterior fossa ependymoma of the type A, supratentorial ependymoma, relafused, here the posterior fossa ependymoma of type B. Crucial to this slide is this line down here. Here you see the indication whether a tumor included in this plot is a primary or a recurrent ependymoma. The trick is, here we always included pairs of tumors, a primary tumor and then later, at a later point, a recurrent ependymoma. There may be 2 or 3 years or even more between the primary and the recurrent tumor, and there may have been progression of the tumor to a higher malignancy. But unsupervised clustering in virtually all cases places the primary tumor next to the recurrent, next to the relapse. Primary tumor brown, relapse here bluish. And in no single instance, the relapse crossed the border to another group of ependymoma. So even if they were not positioned next to each other, they were within the same group. So this unsupervised clustering is a very good practical example that the methylation pattern, as it is looked at in our approach, is extremely stable over tumor evolution and during progression, and therefore suitable as a basis for classification. Now, as I mentioned, the methylation classification was developed in brain tumors. A considerable amount of brain tumors have been analyzed, and then they were subjected to different tests. Here you see already the basis of a brain tumor classifier in its first version, which is based on 2,800 brain tumors, so-called reference tumors, which were classified to the best of the current knowledge. And what you can see is here that in this TNSE depiction, this is a reduction of the multidimensional data to 2 dimensions. You see that tumors which have received the same diagnosis, like here a glioblastoma, or here a medulloblastoma, here also a medulloblastoma, that tumor with a distinct diagnosis cluster in little clouds, it's like little galaxies. But very important is we don't see anything in between. Meaning that if I have a group of reference cases with a medulloblastoma of the Wnt subtype, then if I have another one and co-cluster, it will fall into this group. It will not end up anywhere else. And this, in a sense, is the basis of the brain tumor classifier, and it's also, in a sense, the basis of the sarcoma classifier, which Christian Kölsche will talk to you now about. I'll hand over.
Okay, thank you, Andreas. Yes, I'm here. So now let me start why we have chosen sarcomas as the next study field for DNA methylation-based profiling. The reason for that is crystal clear, because sarcomas are prone for misclassification, and we have here the typical sarcoma subtypes grouped by age group. And you can see very easily here that under the top 4 diagnoses in each age group, we have unclassifiable sarcomas. And what is also quite important to notice here, we have certain sarcoma subtypes which predominantly occur in very young patients, whereas other sarcoma types, for example, GIST or liposarcomas, typically occur in older patients. The problem, however, for each age group is we have a huge amount or a high frequency of unclassifiable sarcomas. And this was the reason why we have focused our research on sarcoma profiling using DNA methylation profiling. Okay, so here you can see our publication which shows you the reference set of our sarcoma classifier. And first of all, I have to say the sarcoma classifier is pretty well working. However, it is not so sophisticated in terms of the reference composition compared to the brain tumor classifier. You might know that we are dealing with over 100 different sarcoma subtypes defined by the WHO. However, in our current version of the sarcoma classifier, we can recognize roughly about 50 different sarcoma subtypes. So there is a lot to improve, and we will do so in the next months and years. So what you can see here is similar to the brain tumor classifier TSNE plot, that each case, which is color-coded here, clustered in so-called galaxies or clouds, and these clouds are composed of points with the same color, and color-coded for sarcoma subtype. And you can see here, for example, synovial sarcoma cluster together, and then you can see here schwannoma, for example, desmoplastic small round cell tumor. Each of these sarcoma subtypes has a distinct DNA methylation fingerprint or profile, and this can be used for diagnostics. Okay, so you can do this clustering analysis and TSNE analysis. However, you have to challenge your reference set, and one of the first validation analysis is the cross-validation, where you use your reference set to calculate your misclassification rate or your error rate. And by doing so, in our reference set, the error rate was below 2%. So the reference set, which we use in our current sarcoma classifier version, has a very low error rate. Then, of course, you have to test for your reference set for potential confounding factors. For example, sex. So here you can see a color-coded male and female here for our reference set, and you can easily see that in most so-called clouds or clusters here, you can see an almost equal mixture of male and female patients. However, there are some clouds where you only have female patients and some clouds where you have a predominance of male patients. And this is very easy to explain because, for example, here this red cloud is composed of endometrial stromal sarcoma cases, low-grade endometrial stromal sarcoma cases. So it is crystal clear why we just have female patients in this methylation group here. And here, for example, this cluster is desmoplastic small round cell tumor, a tumor which is well known for its predominance in very young male patients. Okay, so what other confounding factors are existing? So patient age, for example, also a factor which we have looked for, and you can see here that we have tumor types which cluster together where the patient age is very low, so very young patients like desmoplastic small round cell tumors, or here in this area, I guess this is the gastrointestinal stromal tumor cluster here, where patients are predominantly of middle-aged or elderly. So there is no age-dependent confounding here. Another very important point is the type of material which you use. So we can say that DNA methylation profiling works extremely well on frozen tissue, but also very well on formalin-fixated FFPE material, which is a typical material which you work with in General Pathology and Neuropathology. So you can make use of your archive to fill up your reference set with FFPE tumor material. And we are dealing here with extremely rare sarcoma types, tumor types. So this is a very, very important and important factor that we can use FFPE material for DNA methylation profiling. Okay, then I guess in 2015, Illumina updated its array platform from 450K to the EPIC array, and the EPIC array now can cover over 800,000 DNA methylation positions. And what is shown here is that we don't have any batch effect if we are comparing the data which were generated by 450K chip or EPIC chip. On the right side, on the right side you can see tumor purity. So the tumor cell content is also a very important confounding factor if you are trying to set up a classifier system. And what you can see here is that we have calculated here the tumor purity by an algorithm based on the DNA methylation data. And you can see that we missed our target of 70% tumor cell content in almost all cases. Ewing sarcoma, for example, has a very pure tumor bulk of almost 100% tumor cells. However, we also have tumors which do typically have a very high amount of inflammatory cells. For example, the inflammatory myofibroblastic tumor. I'm sorry of coughing because I got infected by COVID a couple of days ago, and this is why my voice is still a little bit liable. Okay, so but you can understand that tumor purity is a confounding factor. However, this tumor purity is not a huge or does not have a huge effect on our reference set. However, tumor cell purity still is very important if you use DNA methylation profiling for diagnostic purposes. So if you want to generate a tumor profile, DNA methylation profile for a diagnostic case, you must do all efforts, or you have to do all efforts to increase tumor cell content, to increase the purity. There are different methods which you can apply, for example, microdissection of tumor areas and so on. Okay, so this reference set was characterized in detail by DNA methylation profiling, of course, by histology. So all our reference cases were diagnosed by sarcoma reference pathologists or very experienced sarcoma pathologists, and all of our reference cases underwent molecular profiling, meaning if we deal with or if we have dealt with a sarcoma entity which is known for a translocation or typical mutation, we tried to find these mutations. If these mutations haven't already been detected by molecular pathology in the context of reference pathology. Next, we tested our reference set against a validation set. And the validation sarcoma set was composed of over 300 different cases. It was a wide mixture of different entities and of different patient ages. So we had a couple of cases of very young sarcoma patients or sarcoma children, and we had very a lot of patients also of elderly with, for example, undifferentiated sarcomas and so on. And these cases were tested against our reference set. And surprisingly, most of these validation cases clustered together with the methylation set, or with the methylation class, which was expected. However, there were a couple of cases, especially here at this. Let me try to show you where you have to focus now. Here, these are the sarcoma NOS cases. And you can see here that many of these sarcoma NOS cases clustered to DNA methylation classes with distinct histotype. So one of the very interesting facts here is that we had in our initial under the initial diagnosis of our validation set, no case with the diagnosis of sclerosing epithelial sarcoma. However, or fibrosarcoma, sclerosing epithelial fibrosarcoma. However, almost 5 or 7 cases clustered to this group, and by doing additional molecular analysis, we could find the underlying translocation which define this sarcoma subtype. So this, as an example, illustrates you the power of DNA methylation profiling in sarcoma diagnostics. Here on the right side, you can see, you can see again our validation cohort, and you can see here the institutional diagnosis, and here the number of cases which cluster to the methylation classes here on the y-axis. You can see, for example, that undifferentiated sarcoma here typically got a DNA methylation signature of undifferentiated pleomorphic sarcoma. However, there were also a couple of cases which were unclassifiable. And we can come to this very important point later, maybe in our Q&A session. But one explanation is that a single sarcoma subtype can be composed of different DNA methylation signatures. So, for example, a glioblastoma has not one single DNA methylation signature. There are different DNA methylation subtypes in the diagnosis or in the field of glioblastoma. And the same is also true for sarcoma, at least for undifferentiated sarcomas, for chondrosarcomas, for example, and maybe for many more sarcoma subtypes as well. Okay, now you can see here the TSNE analysis of our validation set together with the reference set. And here on the right side, you can see color-coded grayish the validation cases which got a classifier score or which were classifiable, let's say it this way. And you can easily see that most of these cases clustered in the center of these clouds here. However, if we have now a look on the non-classifiable cases, you very easily get the impression that most of these cases cluster between these clouds in a so-called empty room. So there is a pretty high probability that we are dealing here with sarcoma, at least epigenetic fingerprints which are in this reference set not covered by our cases, by our reference cases. We can discuss this point also later on in the Q&A session, if you like. We also had 4 cases which got a misleading result, meaning we had a crystal clear histological diagnosis, and the classifier result was completely misleading in terms of that we got a result of dedifferentiated liposarcoma in a patient with lipomatosis, in a very young patient with lipomatosis. So this was misleading. However, and this is very important, we can also calculate from our DNA methylation data set copy number profiles. And if you are dealing, for example, with a DNA methylation profile or the DNA methylation result indicating for a dedifferentiated liposarcoma, you would expect an amplification of MDM2. This tumor type is defined by MDM2 amplification and or CDK4 amplification. Luckily, in our case, with a misleading classifier result, the copy number profile was flat, so it was very easily recognizable that the classifier result was completely false or misleading. However, you can see that even if you won't get a classifier result, you still have the copy number profile, and you can extract a lot of very important diagnostic information from a copy number profile. It's very important and very helpful in the diagnostic process as well. Okay, so I have shown you a couple of plots with this TSNE in this TSNE form. This is extremely time-consuming, and you need a lot of PC power to calculate these plots. So it is very unfeasible to make a TSNE plot for each diagnostic case. So we therefore decided to use a so-called random forest classification system to make this process much faster. I'm not a biometrician, so I can't go into detail how this classifier works. However, and this is very important, the classifier is composed of so-called decision trees of thousands, 10,000 of decision trees. And each decision tree calls for a methylation class. However, and this is also very important, the random forest classification or classifier allows not to recognize a DNA methylation profile. Very important. So if you are profiling a tumor entity or a tumor subtype, sarcoma subtype, which is not covered in our current version of the sarcoma classifier, the classifier in almost all cases wouldn't give out a false positive call for a methylation class which it was trained for. Very important, DNA methylation-based classifiers allow not to recognize DNA methylation profiles. Okay, so the classifier then sum up the results of the 10,000 decision trees. And the result is a number, a score between 0 and 1. The row scores are these numbers, in fact. However, if you have a look here on the left side, you see that, for example, lipoma, for example, or other sarcoma types have typically a very low score, whereas Langerhans cell histiocytosis, for example, typically have a very high score. This makes it very difficult to compare DNA methylation scores between these different methylation classes. So this is why we generated a calibrated score with a cutoff here at 0.9, which means classifier scores, calibrated classifier scores above 0.9 indicate for a methylation class call or for a recognition of the methylation profile by the sarcoma classifier. Scores below 0.9 are maybe suspicious for, but to say it hard, are not 100% convincing. Very important. Sarcoma classifier scores above 0.9 are pretty sure classifier called for a certain methylation class. Scores below 0.9 are not as convincing. However, they can also be used in diagnostics. But we can discuss this also in our Q&A session later on. Okay, now, Andreas von Deimling, shall we switch again?
Okay, I will resume. So, well, thank you, Christian, for demonstrating the sarcoma classifier. Now, once we have such a tool, an important step is to distribute it, to make it accessible, and for this reason, we have generated a web page, which you see here. It's www.molecularsarcomapathology.org, and if you of this presentation, I would say this is the line I suggest to take a note of, because if you type this in your browser line, you come to this page, and this page does several things. First of all, it explains to you a little bit more in detail what we are doing. It explains, it defines the reference groups we have been including. It gives you some information about the principles. It also contains a lot of disclaimers, because this is not an approved diagnostic tool, so of course it's for research only. But what this page also does, it provides you with a portal where you can upload your own IDET files. If you are in the position to generate methylation data using this technology which we are using, you can upload your files to this page. What we are asking for is that you give the name of the samples. You have option here. You can give notes. We would ask you for localization, and we would ask to state whether the analysis has been performed on FFP or fresh frozen tissues. Although I have to admit we can see this from the data by ourselves. So, and then there's another very important little box which I'd like to turn your attention to here. If you tick this box and upload your files, you are allowing us to keep the data for further education of the classifier. If this box remains unticked, the data will be purged from the system as soon as the analysis you are uploading the data for has been done. So what is the analysis? The analysis is compiled in the next slide. You receive within seconds, hopefully, an email which depicts this sheet. There are a couple of quality control measures. You will see a classifier score. You will see whether your sample receives a calibrated score over 0.9. If it does so, you receive a prediction, and you also, in addition, in any case, receive a copy number profile, which is allowing you to roughly have information whether you have chromosomal gains or losses. For example, this case has losses on the short arm of chromosome 2, gains on 2, or has here a loss on chromosome, on the short arm of chromosome, I think I can't read it really, chromosome 6. Here, a couple of gains. So this is important because, as Christian has mentioned it, this is completely independent from the methylation profile and from the classifier prediction. These are two independent data sets which work together to help to interpret the data received from a specific sample. This page is used a lot. You can see in the next slide the kinetics of the use of this molecular neuropathology side, but the sarcoma side is a subsite of this one. So they are here. The data are taken together, and what you see here is the uploads per year, and we are right now up here. So by now we receive approximately 1,500 uploads per month. Most, of course, are brain tumors, but there's meanwhile a considerable amount of sarcomas with it. And this is the basis of improvement and of new calculations and of the generation of novel classifiers. In the next slide here, you see, short intermezzo. Here you see where the classifier is used currently. And of course, most uploads still come from Europe as it has been developed here. But United States is uploading a lot of cases. Canada, Far East, South America is starting, small numbers yet, but increasing. And Australia is uploading a lot of cases. The cases which we are allowed to keep for further education are continuously and reiteratively analyzed by TSNE, for example. Here you see a TSNE plot which is based on 100,000 or more than 100,000 tumors. And in color, this is the established methylation classes for brain tumors. In gray, you see probably a couple of sarcoma classes, but also clusters which come from other tumors, maybe from breast carcinoma or from colon carcinoma. And whenever we have a tumor we have a really hard time with, we can cluster it into this system, and we can see which groups it falls into. And quite frequently, especially with brain tumors, where we have the highest density of established classes, we find that a novel brain tumor is falling into the void, somewhere in between, maybe in a small group of not colored tumors, and this is then always exciting because this has a good likelihood of representing a novel tumor entity. In brain tumors, this kind of approach in the recent years has been the major source of the detection of novel tumor entities. We and others have detected approximately 40 novel tumor entities over the last 3 years, and the vast majority have been detected by analyzing methylation profiles of tumors not matching classifier output. So really, numbers matter. What are the next tasks? Well, of course, the tasks which we are up for in the next time is we need to further learn and to update the present classifiers. Brain tumor classifier, this has just been done with the result that the initial number of methylation classes, roughly 90, really has been doubled. By now, we are talking about 180 brain tumor methylation groups, brain tumor methylation groups, many of which we don't yet know what they mean. So, in fact, the algorithm for defining new tumor entities has been inverted. Previously, we looked at the microscope. We checked: do we see groups which belong together, and then can we see similar clinics? Can we observe molecular similarities? Now it's just the other way around. We are coming from a molecular side. We see, here we have a group which shares molecular features, and then retrospectively we look at the pathology and at the clinics. And quite astounding is that we find something which we did not expect. By going pathology first, we have now for 100 years selected against brain tumor entities with a high morphologic versatility. Now we recognize that a considerable amount of novel tumor entities are morphologically quite heterogeneous, but share a typical methylation profile and also frequently share typical mutations, typical point mutations in characteristic genes. And of course, what we also find is that several of these novel tumor entities are so rare that they usually are evading a standard approach by guided by the memory of a pathologist saying, I have seen something like this before, let's see whether I can retrieve this and call my friends and find some more of them. Now we are looking at the TSNE and other analysis, define new groups, decide this is interesting, this could be something new, and work from there on. I'm coming to wrapping up, but I'm convinced that this approach, and this also beyond the methylome, will be actually the way how we in the future are expanding our knowledge on brain tumors and on other tumors. I would like to thank now the participants in this endeavor. So for the brain tumor and also for the sarcoma classifier, many people have been absolutely essential. The core group in Heidelberg, of course, is our partners in pediatric neuro-oncology, Stefan Pfister, David Jones, Martin Sill. From our group is myself, Daniel Schrimpf, Damian Stichern, and Christian Kölsche. We have for the sarcoma classifiers many partners in pathology, and many of them are really highly outstanding specialists in the field of sarcoma pathology. And of course, we have a lot of clinical partners. There are many more. We couldn't fit them all here. We hope that this brief introduction in methylation-based classification, using the example of brain tumor and sarcoma classifier, has been interesting to you, and we are waiting now your questions. Thank you very much.
Dr. Deimling, thank you very much for that wrapping up there, and I want to thank, too, Dr. Christian Kölsche for a wonderful presentation outlining the sarcomas and the methylation considerations for classification. And also, Dr. Kölsche, we wish you a quick recovery and applaud your heroic use of your voice today. It's much appreciated. We do have some time now for questions, so let me get right to it. As you can imagine, we always have lots of questions around fixation methods. So we have two around FFP fixation. Does the age of the block or do fixation differences, I'm assuming in time, etc., do they affect methylation profiling? I'll ask you, Dr. Kölsche, to begin answering that question.
Okay, so the typical FFPE fixation doesn't change DNA methylation at a huge extent. So for DNA methylation profiling, it has no huge negative effect. What could affect the quality of DNA methylation is decalcification. So if you are using a very aggressive decalcification system, then you can probably destroy your DNA, and therefore you can't use this material for DNA methylation profiling. However, FFPE or formalin fixation has no huge impact in terms of DNA methylation profiling. The other question was the age of the FFPE material. And interestingly, the age of the FFPE material doesn't matter. So we have used, Andreas, if I'm calling this wrong, you can correct me, but I guess the oldest FFPE block which was used for the brain tumor classifier was 20 or 30 years old. And the quality of the DNA methylation data set was perfect, so it was a very high quality data set, although the material was 30 years old. I hope this answers your question.
Very good. Dr. von Deimling, would you care to make a brief comment about this class of questions?
Well, not a lot to add. Really important is the quality of the fixative. If there's anything which hydrolyzes DNA, like picrinic acid in Bouin, you're done. It doesn't work. But if you use formalin, buffered formalin of high quality, you should not have any problem. What makes a difference is the way we treat formalin fixed tissue differently than fresh frozen. There is a so-called repair kit from a well-known company which we're employing, and this does make some difference. But if you have enough cases, you can compensate in silico for this. You can correct for this.
Very good. Thank you very much. One of the questions that I want to get from both of you an answer. We're blessed today with many new technologies and for molecular testing, and I suppose many in the audience are thinking to themselves, how does the initial tumor testing algorithm for sarcoma tumors and how is the DNA methylation testing incorporated in the picture for other molecular testing like NGS, which I'm sure is very top of mind? Andreas, would you tell us how you integrate those testing methods?
Yeah. I think we really need both. These are orthogonal analysis. The methylation tells you where the tumor comes from, and NGS tells you where the tumor has gone. So for for diagnosis and treatment, you really need both, and we systematically apply both for in our daily routine pathology clinic.
Thank you. And Dr. Kölsche, would you care to comment on that same question?
Yes. Yes. So I think in sarcoma pathology, the advantage of the DNA methylation approach is if you are dealing with a sarcoma subtype which is not defined by a distinct and recurrent mutation. So if you have a sarcoma without a subtype-specific mutation, you can have problems interpreting the result of NGS. If you combine the result you will get from the sarcoma classifier or DNA methylation profiling in general, and combine these information with the histological pattern, the mutational profile, the copy number profile, and the classifier result, all these different layers of information make you very likely a more precise diagnosis. So in terms of sarcoma pathology, this multi-layer approach for diagnosis has especially in cases where you are not dealing with a genetically defined sarcoma subtype. And I think we don't have to think in diagnostic terms that we just use one specific molecular test. I think the future will be that we will use different types of molecular tests, image analysis, AI in image analysis, histological image analysis, and so on, combining these information with the molecular data set, not only dealing with molecular information on the DNA level, but also the proteome might be very interesting. And I think the combination of all these information will make us better diagnosticians.
Thank you. Thank you very much. That's certainly the promise that's being realized as we go along. We have a question here. It seems that the WHO is allowing for tumor classification of CNS tumors by the use of the methylome. Do you believe that bone and soft tissue tumors will get to a same level soon? Or the question is, I suppose, underlying this, do you see any fundamental difference, biologically speaking, between CNS tumors and BST tumors that makes tumor classification by the methylome less feasible? Christian, I'll ask you first.
So, biologically speaking, there is no difference between a CNS tumor or a soft tissue tumor in terms of its malignancy. If you remember the first slide which Andreas showed you, the path a cell goes through from a undifferentiated cell to a differentiated cell. This holds true for all different tissue types. The problem, or what makes it more difficult to build up a sarcoma classifier, maybe is that we are dealing with different compartments in our body. So if you are dealing just with CNS tumors, you are restricted in most cases to the CNS. In soft tissue and bone pathology, you have much more different body compartments you are dealing with. So microenvironment, etc., PP can play a role. So we don't know how DNA methylation of different of different body compartments is. So there might be some difference. However, it should illustrate that it's likely a little bit more challenging generating a sarcoma classifier than a CNS classifier, because you are dealing with just one compartment. We are speaking about CNS tumors.
Thank you. And Dr. von Deimling, would you care to comment on that very same question, the biological difference?
I fully agree. It should work the same, but there's a practical problem. Brain tumors, it's pretty simple to receive tumor tissue where the majority of the cells are tumorous. Sarcoma, also epithelial tumors, we really have the problem of a large stromal portion or of a lot of infiltrates, lymphocytes, lymphocytic hematopoietic infiltrates. And we need for good classification, we need a tumor content exceeding 60%, better 70%. And this something in a considerable amount of instances is much harder to achieve for epithelial tumors or for sarcomas than for brain tumors. With this limit, otherwise, I'm absolutely convinced that it will work also for sarcomas and for epithelial tumors, and that based on this approach in both fields, a lot of novel discoveries will be made in due time.
Thank you very much, Dr. von Deimling. I'll leave that as the last word, and I'll begin to wrap up now from today's very fascinating session. I want to begin, of course, by thanking our speakers, Dr. Christian Kölsche and Dr. Andreas von Deimling, both of the University of Heidelberg. They're doing important work here in sarcoma classification through methylation analysis, and I think we're all intrigued with this. Let me remind you, we'll plan to post the slides and the audio of today's very important presentation on captodayonline.com in about a week. I also, of course, want to thank Illumina. As always, they're in the forefront of the field, and we're very, very grateful for their help and their special educational grant for this program. Finally, of course, I want to thank all of you in the audience. I know we've got busy weeks in the summer, and I'm very grateful and gratified when you take the time to join us for the CAP Today webinars. And so just to sum up, once again, very quickly, our thanks to Dr. Andreas von Deimling, Dr. Christian Kölsche, thanks to Illumina, and thanks to all of you in the audience who've joined us today. This is Bob McGonnagle, and I'll bring today's webinar now to a close.